like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong in my happy place. And I'm right where I belong in my happy place. And I'm right where I belong in my happy place.
Greetings and welcome to the National University of Singapore Yong Lu Lin School of Medicine Healthy Longevity webinar. Thanks for joining us. I want to remind you tonight to use the Q&A function. I actually want to call out the audience because I'm coming to you live from Cloverdale, California, which is right in the heart of the Russian River Valley. I've heard they make wine there, but I wouldn't know about that. I don't think anything is going to get made here this year, though, because it's so hot and dry that nothing's going to survive. But the reason I'm calling the audience out is that it's 4 in the morning. We do this show live at 7 p.m. Singapore time because we love having great questions from the crowd and audience, and we'll be trying to collate those. But I had to get up at 4 for this, so these questions better be good tonight. We have Jan Gruber, who's one of our own, speaking, and I'll introduce him in a few minutes. Before we do that, though, we have one of the members of the Center for Healthy Longevity, Tan Shin Yi, who's talking to us about a recent paper on aging, exercise, and inflammation.
Thanks, Brian. As we all know, exercise is beneficial for us and it can help us to age well. But how does that really work? Well, a study conducted by the Human Performance Lab at Ball State University has an answer to that question. This study, which was published in the Journal of Applied Physiology, investigated how aging and lifelong exercise can influence our immune response and muscle function. The study presented 2 important findings. Aging gives rise to more pro-inflammatory markers in the blood and muscles, which leads to muscle wastage and poor immune response. However, this could be prevented by lifelong exercise. The results shown here demonstrate how pro-inflammatory markers in the blood increase with age, and how lifelong exercise can prevent that. Researchers assessed inflammation levels at rest in 3 groups of men. The first group consisted of young men who exercise regularly, the second, old healthy men who do not exercise, and finally, old men who exercise regularly for the past 50 years, also known as lifelong exercisers. Although aging increased baseline concentrations of inflammatory markers in general, older participants who exercised regularly had significantly lower levels compared to those who did not exercise. Next, the researchers examined changes of pro-inflammatory gene expression in muscles before and after exercise. These graphs show how the expression of pro-inflammatory genes in muscles change with age and exercise. Regardless of age, exercise causes stress to the body, thus an increase in pro-inflammatory gene expression is expected. Muscle tissues from old non-exercisers showed a 2-fold increase in pro-inflammatory gene expression compared with lifelong exercisers after a workout session. Yet, the lifelong exercisers showed similar response to the young exercisers. Exercise also produces molecules, namely interleukin-8, which recruits immune cells to site of exercise-induced stress. The findings showed that IL-8 levels increased 2-fold in old non-exercisers compared to lifelong exercisers who showed similar response to the young exercisers. As aging can impair the immune response to exercise, this suggests that the untrained aging muscle was less prepared than muscle in trained participants to tolerate exercise stress. Thus, we can see that lifelong exercise enables muscles to tolerate exercise-induced stress. Researchers also examined anti-inflammatory gene expression in muscles before and after exercise. At baseline, lifelong exercisers generally showed higher inflammatory gene expression, especially in the molecules IL-10 and TGF-beta. And these molecules suppress the expression of pro-inflammatory molecules and help to modulate the body's immune response. After a workout, there was a twofold increase in TGF-beta observed in old untrained muscles compared to trained muscles in old and young participants. This suggests that lifelong exercisers were better able to adapt to the exercise-induced pro-inflammatory response. Our inflammatory profiles in aging can impact overall muscle health. As shown in this graph, lifelong exercisers were observed to have lower circulating IL-6 levels. They were also observed to have higher skeletal muscle mass compared to old non-exercisers. In conclusion, although aging dysregulates the immune response to exercise and gives rise to muscle loss, lifelong exercise is anti-inflammatory and preserves muscle health throughout aging. Thank you. Back to you, Brian.
Thank you, Shani. Thanks a lot. Jan, I don't know, it seems to suggest that if you're a non-exerciser, you better stay on the couch.
Like that idea.
Actually, it's a pleasure to have Jan Gruber here. He's an associate professor at Yale-NUS and also at NUS Biochemistry Department, where I am as well. He's been in Singapore for a number of years. He's worked on first with Barry Hallowell, but now as an independent faculty member for quite a long time. And he specializes in aging and pharmacological interventions. I think what's exciting to me tonight is he's going to tell us a little bit about one of the model organisms that study aging, and even better, he's channeled Monty Python for his title, which is "What Have Worms Ever Done for Us?" I changed it to us, Jan, because it just sounds more Monty Python.
That's fine. Okay, so I start sharing?
Yes, please.
All right. So yeah, you asked me to talk a little bit about model organisms, in particular C. elegans, and I'm going to do that. So that means going back to the 1990s, but I'm going to start out going back a little bit further in the past. And in particular, I'm going to go back to about 4,000 years ago. But don't worry, it'll not be a linear travel through time. I'm going to start with the Epic of Gilgamesh, which is considered the first example of human literature that is non-religious text, like a story. And it tells the story of Gilgamesh, the king of Uruk. And among other things, he is concerned with the search for the secret of eternal life, which in the end, he basically decides to give up. So he decides not to be immortal. And the thing about this that I think is interesting is that basically the first example of a written text, other than a religious text that we have, asks the question, why do we have to grow old? Why do we have to age? And arguably, for most of the next 4,000 years, the answer was pretty much on the level of the answer of the Gilgamesh. There wasn't really something other that you could say about it, other than that aging is just how the gods distinguish the living, the gods and the mortals. But of course, today it's a scientific question: why do we age? And so when we ask this question, we can answer the question in many different ways. Mechanistically, what kind of macromolecular damage occurs? We can ask, organismally, what diseases occur, what happens. You can, even approach this from a perspective of physics and dynamical systems. But there's a saying in biology that says nothing makes sense except in the light of evolution. So the question is, how does a trait like aging, how does aging evolve, and how does it occur across the biological kingdom? And an interesting point about this is that we have to explain quite a lot variability in lifespan, right? So if you want to look at life expectancy or lifespan or aging rate across animals, you need a measure. And the most appropriate or the easiest way to define aging is as an exponential increase in mortality. So we all know that the risk of dying goes up as we go older, but in fact, it goes up exponentially in a very well understood law. So in humans, every 8 years, your risk of dying in any given year doubles. So that is known as the mortality rate doubling time. So it goes up exponentially, starts out very low at age 1. Then puberty happens, which causes a sudden increase in the risk of dying because of reckless behavior. And then following that, there is there is a clear exponential increase in mortality, which in a logarithmic plot looks linear. So there's an age-dependent exponential increase in the risk of dying. And in humans, the characteristic time step for this is 8 years. But this is very variable across the animal kingdom. So in some whale, it is about 20 years. And in mice, it's about 3 months. And in C. elegans, the animals I will talk about today, it's only 3 days. So what this tells us is that the rate of biological aging is vastly variable across the animal kingdom. There's about a 2,500, over 2,000-fold difference in the speed with which animals age. And from an evolutionary perspective, this then raises the question, how does this get selected? How does this evolve? And arguably, the first group of people that approached aging from a scientific perspective were the evolutionary biologists of the 1930s and 40s. And this culminated in a famous lecture by Peter Medawar in 1951, which was called by Aging, an Unsolved Problem of Biology. And in it, basically, Medawar asks the question, how can this trait aging, the different aging rate, actually evolve? And the important thing about this is that if you want to talk about the trait evolving, you need to look at the conditions under which the traits evolve. You can't make observations from your environment, from human society. You need to look at the wild type situation. So if you look at a mouse in a lab, which is equivalent to our life today in a very protected environment with secure access to food and possibly SPF, so no diseases and no infections, they live about 40 months. About 90% of them die at about 3 years of age, 36 months. However, if you look in a field, you look at field mice and you do a sort of recapture study, what you realize is that life expectancy is far, far shorter. So nature rat in tooth and claw applies, and they die of things very different from old age. They die from infection, predation, accidents, environmental impact, lack of food. And in fact, 90% of them are dead after 9 months, and almost 1 in 2 doesn't really survive beyond 4 months. So what that means is that most animals in the wild probably don't experience much aging. So they die from extrinsic causes. And so this was one of the key realizations of Medawar based on work by Haldane, that aging is really driven by something early in life and not by something late in life. What this means is things that happen back here cannot be selected for because there are essentially no animals left in the wild for selection to act upon. So all of the selective pressure has to be here. Now, the important thing about this is that it led to a sort of particular idea of aging that essentially suggested that aging was a matter of neglect, that it was maladaptive, which is true, but also that it is essentially random and not under some sort of coherent regulation. So, for instance, the typical example is Huntington's disease, which is a human disease that affects humans in late life or later in life after they had children. And because it only affects people who already had children, it can't be selected against, and it will persist in the human population. So the idea was that aging is like that, and that therefore age-dependent diseases are independent and not really under some sort of central control. So the idea is, for instance, that DNA damage repair isn't perfect, so mutations accumulate over time, so you get cancer. Lipid metabolism produces some sticky species, and inflammation is ripe, so you get cardiovascular disease. Proteostasis isn't perfect, so proteins crash out in your brain, and you slowly accumulate plaques, and you get Alzheimer's or Parkinson's disease, and so on and so forth. And if this was the whole truth, then the problem is that there would be almost nothing we could do about aging, because aging would be the consequence of many independent random processes of neglect, of maybe trade-offs where it's just not necessary to make an organism to last. And so basically it's just falling apart, wear and tear. And there's very little we could do about this in this case, because there were many independent causes. Now, the reason why we know that that isn't the whole truth really has to do with these model organisms that I'm going to talk about today, called C. elegans. So these are introduced to science by 1965 by Sidney Brenner as models for development and molecular biology. They're called soil-dwelling nematodes, although in truth they probably live more in decomposing plant matter, like rotting fruit and rotting leaves. They're very tiny. They're about 1 millimeter long, 100 micrometers wide, so maybe like a very short eyelash or a comma at the end of a sentence. They are 99% self-fertilizing hermaphrodites under normal conditions, which means they make copies of themselves, genetic copies. They do not normally make males, although they can. They have a huge proliferative capacity, 300x per adult in about 8 days, which means that they probably live in a really dangerous environment because it suggests very low survival. And we know more about worms than we know about most other animals on the planet, maybe other than people. We know that they have exactly 959 cells and 302 neurons. They were the first organism to have its genome sequenced. They are fantastic molecular tools, a huge amount of knowledge. We know the whole connectome of the nervous system. So this is a little video from my lab, which shows the neurons in the head, basically, of a Parkinson's disease model worm. And it's just, you can image their transparent, so you can image the neurons, and you can essentially watch the neurodegeneration. So they're really good tools for all sorts of biological investigation. And they told us a lot about biology. They usually need the help of some humans who then get Nobel Prizes. So there was a Nobel Prize for the discovery of programmed cell death, which is something we didn't know happened until we saw it in C. elegans. And the same is true for RNA interference, which we only know about because C. elegans told us about it. Now, the worms do not win Nobel Prizes because you have to be alive when you win the Nobel Prize, and they're very short-lived. So the worms age, and they only survive for about 30 days. So here's the lifespan curve. On the left is their percentage survival, 100%, start out, and after 30 days, they're all dead. And the videos on the right show different life stages, a little bit sped up. So the day 6 worms are doing wormy things, crawling around, having fun. The day 16 worms still move a little bit, but really are getting very sluggish already. Some of them are dead. And by day 23, the wild-type worms, the majority of them are dead, and some of them are still, hanging on, but basically they're on their way out. And at this point, I usually say, and in the wild, again, to just confirm that the same principle applies for the mice, in the wild, their survival is far, far shorter. They have a median survival of 1.5 days per day. So they really rapidly die out, which you should have guessed from the fact that they produce 300 eggs. So it's extremely dangerous environment that they live in. Now, at this point, I normally say, this is why they're great models for aging research. But actually, when the first discovery of aging mutants in C. elegans occurred, people didn't really think there was a need, or the majority of people, I guess, thought there wasn't really a need for this kind of research. Aging wasn't really under some sort of genetic control centrally. There were no master regulators of aging. So it wasn't a huge industry of explaining aging on a genetic level. It was basically just seen as an inevitable consequence of this lack of selective pressure for late life events. And so this really was a paradigm shift. This was an important discovery that there are, in fact, aging mutations. So this is a famous paper from 1993 by Cynthia Kenyon. And basically it says a C. elegans mutant that lives twice as long as wild type. And the data shows exactly that. You have a worm, control worms. These are the white squares. And basically, like I just showed you, they're dead after about 30 days. And then you have a mutant that lives twice as long, where there are some survivors up to beyond day 60. This is a very significant difference in lifespan for an organism. There's not normally people or animals that live twice as long as the mean of the population or the maximum of the population. And importantly, it's a single gene mutation. So it is a single position or a single place in the genome where there's some changes. It's only one gene. It's not a selective thing where over long periods of time, like in dogs, for instance, there's selection for longer and shorter lifespan. This is something where breaking a single gene changes lifespan dramatically. And that was unexpected from the perspective of this evolutionary and medical understanding of aging, which was essentially that it wasn't under some central control. And the introduction of the paper says this very clearly. This suggests that there's a regulated lifespan extension mechanism, which was the new idea. And if you look at these guys, this is taken with the wormbot in my lab. On top, you see the wild type, and they will run out of speed. This is over like 20 days or so now. They're starting to die. But if you look at the bottom, the DAF2 animals are still quite happily, scooting around doing wormy things, where there's like one survivor on the top, and the DAF2 worms are still out doing their thing. So they're not hanging around sick and tired and unhealthy for longer. They are just younger for longer. So, looking at the top, everything is dead. There's like maybe one or two still moving their head. And at the bottom, there's still animals literally traveling around, eating and doing their thing. So there's a really dramatic effect with these single gene mutations, which wasn't entirely expected. And now we are at day 50 or 60. And so the last animals are dying. So this immediately brings up some really important questions. So one is, of course, mechanism. So what are the genes? What's the pathway? What is mutated? What changes? How does it work? And I think that's something which maybe we are not thinking about enough nowadays, the cause of aging. What is actually happening beyond the regulation? What is the downstream effects? And it's a hard question. And then the very important question is it private or public? By which we mean, is this mechanism the same across animal species? Is it just a worm thing? Is it just C. elegans are weird, they're so short-lived, and so they have this mechanism, but other animals don't work that way? So is this pathway conserved? Does it control the lifespan in other animals? And importantly, can be translated? So can we make drugs targeting these pathways potentially to extend human healthy lifespan, prevent age-dependent diseases? Now, I'm going to very briefly talk about this because it's a 20-minute talk. I don't have time to go through the whole history of this. But, worms are made literally or born at least into science to answer the first question, mechanism. So it was very rapid through beautiful work by a lot of people that this pathway was identified and mapped, and the pathway elements were identified. And we now know that this mutation is the DAF2 mutation, that the long-lived mutation is the insulin IGF receptor. So it's the receptor that senses insulin-like growth factor. And the important downstream gene that is the transcription factor is DAF16, which in humans is called FOXO, which is a stress response and sulfate involved transcription factor. So, and we know quite a lot about the pathway in between. So, that is something that model organisms are perfect for. And so it was rapidly, solved. What we also know is that this pathway is actually quite highly conserved. So this is a famous slide where they compared the different examples of this pathway in yeast, worms, flies, mice, and humans. And the general architecture of it and the genes are quite conserved. Importantly, mutating this will extend the lifespan in most animals. So it works in mice. If you mutate members of this or elements in this pathway, you get lifespan extension. In flies, you do in worms. And in yeast, it's involved in lifespan extension. How about humans, right? Obviously, we can't really make mutant humans to figure out whether this works. But there's quite good evidence now that it is also involved in lifespan determination in humans. One of my favorite ones is the sort of GWAS studies, where they look at genome-wide association studies trying to identify genes related to longevity. And these are very big, expensive, difficult exercises, looking at large cohorts of humans, 6,000 longevity cases, and 3,000 controls. And this is a meta-analysis. And the funny thing is that what you end up getting at the end is that 15 to 25% of longevity seems to be genetically determined, but only 2 genes show up consistently. And one of them is ApoE, which is known to be involved in cardiovascular disease and neurodegeneration, and FOXO, which is DAF16. So basically, the worms already told us 50% of what we learned from GWAS studies about aging. So I think that's amazing. So that it suggests that this pathway is very well conserved in people. There's also evidence that variations in the IGF pathway are involved in supercentenarians. That is, it changes your probability of living to 100 or to, actually to 100 rather than supercentenarians. So your probability of surviving to 100 sort of depends on your IGF state. So there's quite a lot of evidence that this pathway is involved in determining human longevity. And there's also interesting examples from people who have mutations in the IGF pathway naturally. It's a form of dwarfism called Laronde syndrome. And what's interesting about these people is that they are essentially IGF mutants, and they're small in stature, and they have disadvantages. There are problems to this. And they're not exceedingly long-lived. There are not many like 100-year-old Laronde syndrome people. But what is interesting is that they seem to be largely protected from both diabetes and cancer, even though they don't follow necessarily a healthy lifestyle, and even though obesity is prevalent in this cohort. So in other words, there's an unexpectedly low rate of cancer and diabetes, and the idea is that these are two age-dependent diseases that might be directly linked to the IGF pathway and IGF mutations. So where does this leave us? It leaves us with the idea that age exists as a system state. There is an age of the organism, and it's not just random and independent event. There is a sort of central process that controls aging, but it manifests in different diseases. So cardiovascular disease and cancer and Alzheimer's disease and Parkinson's and kidney failure are all symptoms of aging. They are manifestations of the fact that you're old, you have age, and your age looks like cardiovascular disease. And of course, it is driven by a range of aging processes, senescent cells, oxidative damage, protein aggregation, all the processes we know, they all feed into the aging state. But importantly, upstream of these processes are conserved stress and nutrient sensing pathways that are able to modulate in a coherent fashion this set of aging processes. So what this means is that it should be possible to target these processes to then target age-dependent diseases without having to address an individual disease individually, but rather you address aging directly. So that's what worms have told us, or model organisms have told us. And it's worth just summarizing the paradigm shift, in my view. So 60 years ago, we thought that aging was hopelessly intractable. 30 years ago, we know that in model organisms, there were evolutionary conserved pathways that could change lifespan and modulate aging. 10 years ago, we figured out that these pathways are in fact highly conserved in mammals and humans. And I didn't go through it because it's a 20-minute talk, but there's a large amount of pharmacological drug modification targeting these same pathways, and there are more pathways than just IGF now. Of course, mTOR, I already mentioned, that can modulate the same process. So obviously, this is a very different view from the wear and tear view 60 or 70 years ago. It's a fundamentally different way of thinking about aging, and it is driven by these discoveries in model organisms, the elegans. So what does that mean? Can we have longer living through chemistry, better, longer living through chemistry? Well, one of the really exciting projects that looks into this is the ITP intervention testing project by the NIH National Institute of Aging, which looks at lifespan in mice and tests these interventions. And it's one of the reasons why we know that drugs can extend the lifespan of mammals. But they have tested on the order of 6 compounds a year since they started, because mouse lifespan studies are slow and expensive and difficult. And, especially if you do them well, they take a lot of effort. So how can we do drug discovery for aging? What do we want to screen? Well, we might want to repurpose FDA-approved drugs, and there are 1,500 of those. So there are a lot of FDA-approved drugs that we could target. We might want to look at supplements or compounds called GRAS, generally regarded as safe, supplements, basically. There are compound libraries from drug companies that are tens or hundreds of thousands of compounds large. And of course, almost anything will ultimately work as a combination, a cocktail, a stack. We all sort of know multivitamins and multi-everything interventions. And so that makes the search space even bigger. For instance, if you wanted to look at every pair of 2 FDA-approved drugs, you have 1,500 FDA-approved drugs, there are over 1 million pairs. So at 3 or 6 or 10 or even 50 compounds a year, you're not going to get through the search space. You can do hypothesis-driven confirmation. You identify an interesting compound and then you test it in mice. But if you want to address this sort of search space, really your only choice is worms, robots, and machine learning. So the idea is that you need to automate this process to be able to do thousands of lifespan studies. And this is video from my lab using an automatic robotic microscope to do this. And we can do hundreds of drugs at a time with this. And that then produces, on the order of 25,000 pictures, which then you need to analyze. So of course, this needs to be automated. And we do this by, training a neural network that then classifies worms, counts them, and turn this into lifespan. That produces survival curves, a really exciting, and one of my PhD students is working on this, Diogo Barado. One of the really exciting projects recently we got is a worm bot, so 2 worm bots, which is a different system developed by the Kerbeline lab. And that one gives you much higher resolution data on these cohorts, and it can do 144 cohorts per robot in parallel. So there is a huge amount of technology that we have to do very fast parallel lifespan studies. But at this point, you'll probably say, well, okay, fine, you can do these giant lifespan studies, but how do you know it applies to us? How do you know if it works in worms, it doesn't work in people, right? So since I get these questions every time, I went to the ITP, to the Intervention Testing Project database, program database, and I took every drug they did in mice, and I tried to find a worm equivalent lifespan study. And about half of them we did in our lab, and the other ones I found from the literature. And if you plot the lifespan extension in the worms against the mice, so here we have mouse lifespan studies and the y-axis, and worm lifespan study on the x-axis, I think it shows some really interesting stuff. So first, it shows that the zeros really stack up. So I use jitter here so you can see how many there are. But fundamentally, the compounds that the ITP tested and that failed in the ITP also failed at least in our worms. So, the worms told you that this is not going to work, and it didn't. The other really interesting thing that I found was that the best single lifespan extension, I used the largest lifespan extension in the ITP, is actually metformin with rapamycin. That's an anti-diabetic together with rapamycin. And so it's a combination of 2 things. And the fascinating thing about this is if you compare the lifespan curve, so this is rap plus met, rapamycin plus metformin in my lab in worms, and you compare it to mice, you get almost exactly the same thing. It looks very similar. It's a small additional effect on rapamycin, on top of rapamycin. So adding metformin to rapamycin does only a little bit extra. But fundamentally, the sort of magnitude and in some way even the shape is similar. And we also find that while the lifespan extension in magnitude isn't that big, there's some benefits of having metformin with rapamycin, and the same thing has been found in the mice. So I think the worms are pretty good at least enriching your screening pool for the mouse studies. So that made us think we want to do a systematic screen of drugs, drug-drug synergies, interactions between drugs. And so we started with a very small thing, this before the robots manually. It was done by another one of my PhD students, then now a postdoc. And what we did is we screened, sorry, I went back. What we did is we took rapamycin, rifampicin, this is an antibiotic, metformin, which is an anti-diabetic, psora, calcium channel inhibitor, and allantoin, an oxidation product of urate that we were interested in. And we made every possible combination between those 2. And I'm going to make this very short, but this is the interaction I just showed you, where the blue is the rapamycin plus metformin. And with this very tiny screen, just 10 pairs, we actually started treating at day 6. So we're treating adult animals. We found quite a dramatic additional lifespan benefit. So rapamycin plus rifampicin plus allantoin almost doubles mean lifespan and increases maximum lifespan by 50%. And so this is a very dramatic effect. And it's a tiny screen, right? We went and confirmed that in flies. So here also rap plus rif and rap plus rif plus allan are very efficacious in the flies. And that means if you look at the evolutionary tree up here, look at the phylogenetic tree, fly and nematodes, flies and worms, had a common ancestor about 1 billion years ago. So these are ancient pathways. I always make the joke that certainly, rapamycin would extend the lifespan of T-Rex if you tried it, because that's only 65 million years ago. So this is a very ancient conserved mechanism, and even the interactions seem to be conserved. So, but importantly, does it make them happy? Are these happy worms? Well, here is just a video showing you 23-day-old untreated and treated worms. And the untreated worms, you already seen, they're mainly dead. Some of them are still slightly moving. But the treated worms are still essentially happily running around and doing their wormy thing. And I think I know which one I would like to be. If you count that systematically, what you find is that while the treated animals live far longer, they also spend a higher fraction of their life in good health. So the controls spend less than half of their life in good health in this green category A, whereas the treated animals spend almost 60% at optimal health. And that's 60% of the extended lifespan. So they live longer, and a larger fraction of the longer life as well. They are younger for longer. To put that yet another way, the untreated animals spend about 9.5 days in good health and then 10 days in ill health. The treated animals have 21 days of good health, more than twice as much. And then there are 13.6 days that they're ill health in this study. So there's a slight extension of the less well days, but it's less than for the well days. So there's a relative compression of morbidity. So, I think this is, very exciting. And if you want to read more about it, you can read the paper, which Tess published. But the important question is, what is the take home here? So this worked, and it's interesting, and we're doing mechanism and all this stuff. But if you go to Drug Age, which is a database of compounds that haven't been shown to extend lifespan in at least some animals, you find over 600 compounds. So should we just all stack them together, like scoop them in, make a mega anti-aging pill, stuff them all together and take them all at the same time, because, the effects will just add up or synergize. And, having worked in an antioxidant lab for a long time and having seen many of these things not work the way you'd expect when you combine things, we were quite worried about this. So we did a larger study, and that's unpublished yet, but we are about to finish it, where we started with about 17 single drugs, which gives you 136 possible pairs. And then we screened about a third of the possible pairs using this robotic system. And what we found is really interesting. So it's almost exactly a third and a third and a third. 1/3 of combinations are in some way beneficial. Only about 18% was synergistic, meaning that the effect was larger than the sum of the individual effects of the drug. And that's usually if one individual drug does very little and it then does more in the combination. But only a third of them are beneficial at all. A third of them are detrimental, and in fact, 10% are outright toxic. In other words, they kill the animal. They live shorter than wild type. And so what this means is you really need to test interactions of drugs before you start putting them together. So saying, I read these 15 papers and they all have really cool compounds and I'm going to take all of them, which I guess we are all prone to doing, is dangerous, because you don't know whether the interactions will be beneficial or detrimental. You have basically 1/3 chance that they literally cancel each other or be worse. So maybe no mega super 50 everything anti-oxidant anti-aging pill yet. Finally, of course, we're doing mechanism, and what we want to do is eventually explain the synergies. And we just have to look for time. I think I'm out of time, so I think I'm going to stop there.
I expect Germans to be on time. I don't know.
How much did I run over? Sorry.
I'm not telling you. Just everyone, we're going to run the show about 5 minutes late, just so we have more time for questions. And I just wanted to say, you also brought up Bob Horvitz, who won the Nobel Prize for defining program cell death. He was on my qualifying exam for my PhD, and I'm still in psychological counseling trying to recover from that. This guy was one of the sharpest guys I ever came across, which is great 98% of the time, but maybe not so good in your qualifying exam. You started out as a physicist, and you're like the fourth person on the show that was a physicist. What is it that brings all these physicists into aging?
I guess they think that they can do everything better than the biologists. They discover at some point, I'm not saying it's true, I'm just saying they discover at some point that aging is interesting, and they think all that the biologist needs is another physicist to solve it.
Very, very honest question, a very honest answer. I think they do bring a lot to it, though, because of a different way of thinking about the problem than the biologists do. I mean, we're trained in a very non-quantitative way, and I think that it's a good compliment to have people that are coming in from a different perspective, thinking about the problem. And a lot of people in the field started out in the physics world. All right, I'm going to ask the question: how much of it is really conserved in worms? I mean, of aging, there can't be every pathway. There must be pathways specific to worm aging that don't affect human aging and vice versa. Can you comment a little bit on that?
I'm sure there's. I mean, most obviously, I guess, is statins, right? I think simvastatin didn't even work in the mice. But how much heart attacks do mice get, and worms don't even have. Cardiovascular disease. So I think there's certainly things that are private to specific organisms. There's no question about it. I think the high level stuff, like nutrient sensing stress response, obviously turns out to be highly conserved. And actually, I didn't really go into this, but while we have mapped all the upstream, there are 2,000 or so things that are activated by FOXO or controlled by FOXO, right? So the idea that you can then look downstream and figure out whether oxidative damage or lipid metabolism are crucial for aging, it gets really difficult because you're really in the weeds there. So I think the upstream is probably conserved. The details of the downstream are probably not. So it depends where you're targeting. But I think it's surprising the drugs work, right? I mean, why would the drugs consistently preserved?
I mean, if you look at things like nutrients, the pillars or hallmarks of aging, things like nutrient signaling, metabolism, those are, I think it's pretty easy to see those as being conserved. But let me throw out a couple others and get your comments. How about cell senescence?
Well, I mean, so worms don't really have, they're post-mitotic other than the germline. I don't think there's been much work on senescent cells in C. elegans. So I would be surprised. I mean, I would not pick them as an organism for SASP, right? That would not be my choice. And that's why I haven't done much on that. And if what we have done, the little bit we've done in tissue culture. So I think that's probably an example of a mechanism that's more prevalent in organisms that have tissue regeneration.
How about inflammation? I mean, there's an innate immune system. So.
Yeah, so I think that's probably, actually, I think that's sort of underappreciated because they respond quite a lot to the bacteria that you culture them on. And some of that probably has to do, even links back to oxidative damage. So when they respond to bacterial infection, they produce a robust response that involves and includes production of free radicals and increases damage. So I think there is an aspect of this in worm aging that's conserved. Whether that's, again, maps perfectly to people is another question, right? I think the mechanistic thing downstream is getting really difficult. But for instance, I'm just actually really surprised that the sort of interaction between mTOR and IGF, that genetic synergy and the drugs seem to be doing the same thing. For instance, works in worms and flies, right? I mean, even that, there's no reason why this shouldn't be a weird fluke in worms. It just suggests that this is ancient, right?
Yeah, I mean, even yeast, a lot of these pathways are working.
Exactly.
Why is combinatorial so difficult? I mean, and this maybe comes back to the mechanism question a little bit because, it's been, we have studies in mice that are very similar. I mean, you really can't predict what's going to be synergistic or antagonistic, and when you start putting things together, and it makes me nervous when I see all these multi pills out there, poly pills targeting aging, etc. I think that it's we just don't know enough about it, but I don't know why it's so hard to figure it out. Well,
I mean, it's a non-linear complex system, and you're talking for like a physicist, and you're pushing it in one direction and another, and you're expecting that the end state of the two things, of doing two things at the same time, will be the sum of the individual interventions. So we did this with gene expression, right? When we did the drugs, you can do this very easily. You put a drug on, you measure gene expression, put another drug on, you measure gene expression. And then you essentially say how much of the gene expression changes that I see when I put both drugs on together can be explained as just adding the two drugs and even allowing fitting, so a linear combination of the two individuals. And it's not a lot. So the response of putting two things together is qualitatively different than adding one and then adding the other, in post. So they're not linear combinations of the individual interventions, even at the level of transcription. And on top of this, we don't know which of the 2,000 transcriptional changes are directly related to aging, right? We haven't sorted that out yet. I mean, I didn't show the last slides, but this is why, this whole work on clocks is so exciting, because you might be able to sift through these transcriptional changes and assign them to causality, right? To say, okay, so this affects clock modules, and therefore this is the reason why this works better than this. And then you might be able to get somewhere. But I think fundamentally, with transcriptomics getting cheaper and cheaper, at some point it might make sense to do that before you even do the lifespan for the combinations.
Yeah, a biology way of thinking about it is that these drugs are hitting certain pathways, and that if you have two drugs that hit the same pathway, you might hit that pathway too hard.
Yeah.
Or maybe you optimize it with the two drugs and hit it just right. Alternatively, if you're hitting different pathways, you're changing the system in a way that you don't know by hitting one pathway, you don't know what the effect of hitting the second pathway is in that context. And so I think it makes sense. I'm just not sure how to tackle the problem. We haven't gotten to the point where we can predict outcomes yet.
No. There's a little bit of literature that says that when you do sort of drug design for more traditional applications like diseases, you have a disease network, so you have known genes involved. And if essentially the drugs affect dissimilar, non-overlapping, but adjacent parts of the network, in other words, you're like cornering the disease network from both sides, then they tend to have strong interactions, good or bad. And I think that's a measure you can probably sort of make a heuristic for and say these two things are probably going to do something unexpected. By basically saying they're not overlapping, they're not the same, but they're not completely distinct. They do things in adjacent parts of the network. But of course, for that, you need to have a network first, which is where, all this, transcriptomics analysis and things come in. It's not a solved problem yet.
Yeah, I mean, I think it's really challenging. I know that if I can ask a personal question while we're on the topic of interventions, I know you're like something of a hacker. You've tried a lot of things on yourself. What have you learned about testing yourself?
Well, one thing I've learned is that lots of things I was scared about, really, I had no reason to be scared about. So I think it's a weird observation, but if you're relatively young and healthy, for instance, metformin, I've talked to my mother and her friends, and they all claim metformin is the most awful drug on the planet. It causes all sorts of problems. I've been taking it for like almost 3 years now, and I have never had any problems. And I think that's because I don't start out having some sort of metabolic defect, right? And so, I think actually starting early is a really good idea if you can manage the risks, because your likelihood of cascade failure and weird side effects is just so much smaller. It's the same as exercise. That's one thing I've learned. I think that's maybe.
I mean, you could argue the same thing about rapamycin. I mean, the studies on rapalogs in relatively healthy individuals show that you can do it in a way that has pretty low or side effects, not really above background. But, it can cause side effects in people with diseases. And so I think there's a good point there that may be generalizable. Yeah. You mentioned a couple of drugs that people are going to ask questions about, so I'm just going to bring them up. I think people have heard of rapamycin before. Rifampicin.
Yeah, so this is the thing. It really robustly extends the lifespan in C. elegans. And initially, my intuition was that it was probably some sort of caloric restriction because it's an antibiotic. It's an antibiotic given for tuberculosis, and prior to operations, it's like applied to the skin. It's quite nasty, but it's not. So we tried this, and it doesn't matter whether the bacteria are alive or dead, and it works in the flies. So it really doesn't seem to be doing this. From the transcriptional changes in the literature, it might be doing something to junk. It sort of replicates an IGF inhibition in the rapamycin treatment when you look at the transcriptomics. So anyway, it's an antibiotic. I wouldn't suggest people start taking it. It's an experiment. It's, if you figure out the pathway, there might be a safer way of doing this. Allantoin is really interesting because it's an oxidation product of urate. And I used to work in Barry Halliwell's group as a marker of oxidative damage many years ago. Because it's in humans non-enzymatically produced from urate, It's used in skin cream, is mildly anti-inflammatory, very safe. And it surprisingly works as a lifespan extending compound in C. elegans, and there hasn't been all that much work on it. So I actually think it's a very interesting compound. And it's also interesting because if you look at the transcriptional changes, there's very little. It's very few. It's like rapamycin changes 10% of the genome. But allantoin does very little, and still it extends lifespan quite robustly in at least in worms and to some extent in flies. So I thought it was a really interesting drug to look at for that reason. We saw the paper about it and we immediately jumped on it. Metformin, rapamycin, rifampicin, and Psora is a calcium channel inhibitor that we think might be a little bit caloric restriction mimetic. Petrocek's group identified it and he's working on it. And I don't think anybody fully understands how it works. We did quite a bit of work trying to understand the mechanism, but it works very robustly as well. And so we added it to the mix. That was our original thing. And then the bigger screen we just did, we had 17 drugs. I must say, for instance, I'm quite interested in lithium right now, because it does some really interesting things.
Makes you happy.
What are the doses that we use, which is about 10% of what you would use for mood? I don't think it changes mood all that much, but yeah, I think it's very interesting.
Let me ask one more question, then we'll go to Stephen. Are we done with worms and flies and yeast? What can we still learn from them?
Well, I mean, for one, if you want to do anything about this combinatorial stuff, you're never going to do this in mice, right? You're not going to do 1 million mouse lifespan studies, or even if you want to do 1% of all FDA-approved pairs, thousands or tens of thousands of pairs. And don't forget, you have to do this at drug optimization, right? So each of these pairs, you have to do multiple drug combination, concentration combinations. So just to ask the question, what is the, sort of scope, what percentage of things might interact, how do they work, mechanistic things, and then the fundamental question, does it have to do with damage, for instance, right? All of this, I think there's still endless amounts of stuff we can really only do in model organisms. And sometimes, when I get a lot of pushback, especially from the medical community who really feels that if it's not a mammal, it's too far to be of big interest. My answer is that a lot of times the really exciting thing in the clinic was exciting in the worm and fly field 20 years ago, right? So if you want to know where the billion-dollar startup will be in 2040, you really want to talk to worm people now.
All right, let me bring Stephen in, partly because, Stephen is doing a PhD working with me, but he's also working with Jan. And so here's your chance to turn the tables, Stephen, and interrogate the professor and ask all the hard questions.
Think carefully.
Jan has been really good to me in his lab and giving me lots of great advice. Yeah, so I really enjoy working in his lab. Sorry, Brian. I do enjoy working in yours too. Yeah, so Jan, thanks for the talk. It's a great talk. So there have been a lot of questions, and I'm going to try my best to summarize them as best as I can. Yeah, so earlier yourself and Brian were discussing about cellular senescence as an example of translatability. So there's also this question of, when people talk about this field of aging, one of the biggest concepts is the hallmarks of aging, the paper by Carlos Lopez-Otin in 2013. So the question is, do we see the evidence of all of these hallmarks of aging in the C. elegans? So just now you mentioned senescence as one which is not really seen. So what about the rest?
Well, so to be honest, we haven't, I don't think, I might be wrong, but I'm not aware of a lot of work on senescence in C. elegans. I worked for many years on antioxidants and free radicals in C. elegans, and there's certainly an increase in oxidative damage. But one of the things that is one of those conserved zeros is that extending lifespan with antioxidants, like modulating aging rather than age-dependent diseases individually with antioxidants, is really hard and doesn't really work that well in C. elegans or flies or mice or people, right? Like mega antioxidants, super antioxidants, like mega dose antioxidants don't extend the lifespan in any of these species, and that's true in C. elegans, although there's an increase in oxidative damage. Mitochondrial dysfunction is certainly a feature of aging in C. elegans. Protein aggregation is certainly a feature of aging in C. elegans. So many of the hallmarks are conserved. Now, again, cellular senescence, probably less so. Inflammation, you have to be very careful how you define it, right? So there is certainly aspects of this that you could map, but it doesn't have the same inflammatory pathways that humans have. So I think, yes, there are hallmarks of aging that are preserved, and there are others that are not. And I think it comes down to picking the right model organism for the kind of hallmark you're interested in.
Yeah, thanks for the answer. So I'll just go on to the next question. So there's quite a bit of questions on the drug synergy, the results that you showed. So especially the rapamycin and metformin drug synergy, which seems to have quite a bit of potential. So the question is, how translatable is this particular combination of drug synergy from the C. elegans, rapamycin and metformin to humans?
Well, I think this is easy to answer because the intervention testing program did it, right? They did the combination, and it does work in mice. Now, caveat there, it is not clear how much additional lifespan extension over an optimized rapamycin protocol metformin gave in this because of methodological things. But what is much more important in my view is, first of all, in our worms, you can get the same lifespan extension, but with half the rapamycin dose, right? So that would certainly reduce side effects. And there's also evidence now from mice that suggests that some of the rapamycin-related side effects to do with glucose metabolism, for instance, can be ameliorated by metformin. So I think this is absolutely something that we should look at for translation. It works already, both in terms of pure lifespan, but also certainly in terms of dose and how much rapamycin you need, and in terms of functional health and side effects in mammals already, right? So I think, for me, this was a confirmation. And I started this before the intervention testing program results were out, but we knew about it by the time we published the paper. So for me, it was a positive control. It was like, yeah, the worms showed the same thing that you get in mice. You get a small additional benefit. It's statistically significant, but not impressive. But then when you look at it, in terms of other factors, such as how much rapamycin you take, it is very useful. Just a side note, we combined metformin with lithium for in models of neurodegeneration, and there we find a really dramatic compression of morbidity, right? So they don't live that much longer, but they have far less basically detriment. They have almost no morbid period. They live as long as the longest lived treated animals, but they're essentially healthy until they drop. So, I think these combinations, metformin plus rapamycin, certainly are interesting.
Yeah.
And we know the drugs very well, right? I mean, the people take both of those already, so.
Exactly. Looking forward to translatability in the future. Yeah, so there's another interesting question. So I was quite intrigued by this question myself. So this question is it experimentally possible to implement lifestyle modifications such as exercise in C. elegans?
So we've done this. We've exercised C. elegans. It's really difficult. I mean, it was cute. So we put them on audio speakers and played the tone, and that upsets them, and then they run. But if you do this continuously, they habituate. So you have to build a sort of system where you upset them regular intervals. And yes, you can ask the question, is it the stress or is it the exercise? Anyway, so what we ended up doing is making the worms move significantly more. And we have a paper on this, and it works really nicely. It didn't change lifespan. If you do it too much, it shortens lifespan. You can overtrain them. It does change speed, so they are trained, but we didn't get a statistically significant lifespan extension per se from this. So yes, you can train worms. To be honest, I guess there are probably animals that are, more appropriate for that sort of experiment. But yeah, it does work. You can make them fitter, but they don't live longer.
So there's another question about the C. elegans as an aging model organism. So I think that was the focus of your presentation and talk, to show us that C. elegans can be a very useful model for aging. But this question is focusing on how does it compare as a specific disease model. So for example, neurodegenerative diseases, cardiovascular, metabolic syndrome, etc. So
C. elegans, because they're so easy to genetically modify and there's so many awesome tools for them, there are a huge number of very well-made and beautiful models, but they model a specific thing, right? So for instance, we made, and other people made before us, models where human amyloid beta 1 to 42, the pathogenic peptide, the peptide that causes plaques, is overexpressed in neurons. And they show detriments, and you can then treat them and look at what happens. but you have to be clear what you're modeling, right? You're modeling the pathological consequences of making this particular fragment. Of course, you will not be able to model modifications that are going through its production, for instance, right? Like if you change something about the proteostatic system in such a way that you're making less of this, or you're clearing it faster, you're not going to model this by overexpressing it by an external driver and then looking at it. But you can model, for instance, whether you can protect the mitochondria from the detrimental effects by some intervention, or activate proteostasis and degrade it. And that's true for all of this model. There are Parkinson's models for worm. We're working with them a little bit. There are A-beta models. There are lots of disease models. But basically what they do is they model a particular aspect of the disease. And then you will be, potentially it's a very useful model for that disease. Now, another way that we approach this is if you look at endogenous protein aggregation. So you're looking at worm proteins that accumulate over the course of life in worms and cause disease. You can consider this as sort of worm equivalent of AD, right? So they're making some sort of pathogenic aggregates and that kills them. But then you're doing a thing where you have to carefully map the one to the other, right? So the answer is yes, there's a huge amount of stuff we can do, but you need to be very clear about what it is that you're modeling.
The next question is actually a more personal question to you and your lab. So what was the most significant or surprising finding in your lab to date since you started working with worms from a physics background?
Well, so there are two things. So one is I started out at a time when free radicals were the paradigm, right? The eukaryon compounds like catalytic antioxidants, mitoQ. And this was really, that's why I got into it, because it made sense from my perspective, damage accumulation, reduce damage. And so there was a long journey of measuring this and essentially convincing myself that, scavengers or antioxidants will, they have a place in disease, but they will just not change aging rate per se. And so, that this is a sort of negative result, right? It's disappointing. You spend 3, 4, 5 years working on this. And in the end, you give a talk where you're going like, well, we now measure this very carefully and it really doesn't do what we wanted it to do. The flip side of it is that we've actually extended the lifespan in C. elegans by irradiating them with gamma radiation. And, hormesis is a really powerful mechanism. Causing a little bit of additional stress really extends the lifespan of almost any animal. And that's an interesting thing, which I think has to do with those nutrient sensing pathways and stress response pathways that we're talking about. So, reducing stress really doesn't necessarily work the way you want. In exercise, for instance, you can block exercise benefits by like mega antioxidants, right? So that was surprising, if maybe a little disappointing. And then I must say the fact that this synergy, it's a very small screen, right? We're a small lab. It was manual. There was one PhD student. Tess worked on it. Dasale. He did beautiful work, and he got something that almost doubled lifespan in adult animals, right? You wait until they're adult, you give them some drugs, they live twice as long. They're healthier for all their life. They outperform the wild type at every age. So seeing this was really, for me, encouraging, right? At the time, I think the longest lifespan extension in any animal based on adult drug interventions. Depends how you count it. But for me, this was really surprising how easy it was. It was surprising that out of 10 interventions that we picked with some intuition on pathways, this actually worked. It could have very easily been 10 dots. And, Tess was very worried that he would do 10 combinations, then write a paper that says anti-aging drugs don't combine, right? And it's a terrible paper to write. So I'm actually surprised how easy it is. But remember, when you do a larger screen, at least in our next size screen, and it's still only 45 compounds, pairs, 10% are toxic, right? It's not just everything you add somehow adds up beneficially. You have a 1 in 3 chance that they add beneficially, even with those drugs that we pre-selected.
So it sounds like it'd be worth maybe doing the whole 1,500 compound FDA library.
Yeah, and with Matt's robot, the worm bots now, it's no longer, like Tess did about 200 lifespan studies in 4 years as a PhD student. We now can do 200 lifespan studies in 1 month with the 2 robots. And the robots can be scaled. You could have 100 robots, right? It really is, everything is coming together. And machine learning means you don't then have to look through, 150,000 pictures, which was, the bottleneck before. You end up with like 400 gigs of movie file, right? So now the AI will do this for you. So yes, everything is lining up.
So a pathway to identify new aging compounds, maybe.
I think so.
Well, thanks a lot, Jan, for doing this. It was really informative. I'm sure everybody learned a lot about worms, and hopefully they'll get more excitement in that part of the field. I have a few comments to make before we close. First, use the panelists and all attendees button if you want to make comments about the show. We also have a LinkedIn events page that will be posted in the chat box that you can see, and look at that for future shows that are coming up. Next week, we're going to have a new host for a couple shows, Andrea Meyer, who is an expert in aging research. For some reason, she likes to work on humans rather than animal models. I don't really understand why, but she can explain more. She will be having Nick Guldemond on, who's talking about the relationship between healthy longevity and health systems. I'll be back in September with a fireside chat with David Sinclair. So that should be fun and interesting. We were both in the same lab together many years ago, so we've known each other a long time. And I just want to leave you tonight with the comments of Warren Buffett on how to age healthy.
Warren, how would you define true success?
Well, I've said many times that if you get to be 65 or 70 and later, and the people that you want to have love you actually do love you, you're a success. I've never seen anybody that reaches that age. I mean, I'm not talking about somebody that's in extreme poverty or pain or something, but I've never seen anybody that, if they have a lot of people that love them, that is other than happy. And I've seen some very, very wealthy people that they give testimonial dinners to and name schools after and everything. And nobody loves them, you know.
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.